And now we're going to continue this lecture with differentiation of T cells. Okay, so T cells originate in the bone marrow, but they mature in the thymus. So initially, they start as double positive cells, meaning that they express both CD4 and CD8, and then they differentiate into one lineage based on MHC recognition. So if a cell recognizes MHC1, it is going to be CD4, and if a cell recognizes MHC2, it's going to be CD8. Now, we introduced this topic called positive selection in our previous lecture, but we haven't dwelled deep into it. So here I'm going to talk about what exactly positive selection means. And one thing that you have to remember is that it occurs in the thymic cortex, and here the T cells can recognize self-MHC molecules. So T cells that recognize the self-MHC molecules, they survive. So if a T cell cannot recognize self-MHC at all, it dies. Now, this ensures that surviving T cells can interact with MHC in the periphery and is extremely important. Now, let's go to negative selection, and this is opposite of what positive selection is. So if you have a T cell that binds too strongly to the self antigens, that T cell is going to be eliminated. And this process happens in the thymic medulla. And you also have to remember that this is regulated by the ARI gene. So if the ARI gene is defective, the self-reactive T cells are going to survive. And if they survive, that can lead to autoimmune polyendocrine syndrome. And this is very, very, very important for your exams. Now, macrophages present antigen on MHC2 to CD4 T cells, and this has to make sense because MHC2 is present on all antigen presenting cells, and macrophages are antigen presenting cells as well. So when this happens, the T cells they secrete um, interferon gamma, which activates macrophages to kill the intracellular pathogens. And this mechanism is central to type 4 hypersensitivity reaction, and this is seen in TB or tuberculosis. Then you have your CD8 T cells, which target cells through preferins and granzymes. And it's also important to remember that preferins they create pores, whereas granzymes they induce apoptosis. This is antigen-specific killing, and this differs from natural killer cells, which do not require antigen recognition at all. Now let's move on to regulatory T cells. Now the regulatory T cells maintain immune tolerance. They have CD4 positive on them, CD25 positive on them, and they also express FOXP3. And it's also essential to remember that they suppress immune responses using IL-10 and TGF-beta, um, which reduce the immune response. And you also have to remember that the loss of FOX3 causes IPEX syndrome, which presents with severe autoimmunity in infants. Now, this is very important topic for any exams, and when we talk about naive T cell activation, you must remember one golden rule, okay? T cells require two signals to become activated. If you miss either signal, that leads to dysfunction, not activation. So your first signal, as you can see on this diagram, is called antigen recognition. So first look at the antigen presenting cell. This could be a dendritic cell, a macrophage, or a B cell. The antigen presenting cell processes an antigen and presents it on MCH1 for CD8 T cells and MCH2 for CD4 T cells. Now, on the T cell side, the T cell receptor recognizes the antigen MHC complex, and this interaction provides signal 1. Now, the key concept to remember here is that signal 1 tells the T cell what antigen it is seeing, but not whether it should respond, right? For your exams again, I would like to stress that signal 1 is not enough for activation. Now comes your signal 2, which is called co-stimulation, and this is your permission signal. Now, if you look at the second interaction, CD8 is expressed on the T cell, B7 is expressed on the APC, right? So when you have binding of CD28 to B7, it provides your signal 2. Now the key concept to remember in signal 2 is that signal 2 tells the T cell that this antigen is dangerous and it's worth responding to. And this signal leads to production of IL-2 production, T cell proliferation, clonal expansion, and it also causes differentiation into effector and memory cells. So what happens if signal 1 occurs without signal 2? This is one of the highest yield immunology concepts on any exam. So if TCR binds your antigen MHC molecule, which is your signal 1, but no CD28B7 interaction occurs, which is your signal 2, the T cell becomes something called anergic. And anergy refers to functional inactivation. So meaning that the T cell is alive, but it will not respond to the antigen again. And why this matters clinically? This is a major mechanism of peripheral tolerance and it prevents autoimmune responses. Okay, so always make sure you at least get the buzzwords for your exam. Signal 1 without signal 2, co-stimulatory signal is absent, and T cell becomes unresponsive and it goes anergic. Make sense? Let's move on. Now, now we move into humoral immunity. Now, this is specifically called T cell dependent B cell activation. In your first step, you have antigen binding and internalization by B cell, right? So start with the B cell antigen. So antigen binds to the B cell receptor, right? So you have BCR, B cell receptor. The B cell internalizes the antigen. The antigen is processed and it's presented on MHC2, right? Now, important distinction is that B cells are professional APCs only for antigens they specifically bind. Okay, so step 1 is completed. Step 1 is what? Antigen binding and internalization. Now we move on to step 2. Step 2 is when there is interaction with CD4 T helper cell. Okay, now the CD4 T helper cell comes into play. You have something called TCR, T cell receptor on CD4. Now the TCR on CD4 T cell recognizes the antigen MH2 complex on the B cell, and this provides signal 1 to the T cell. But again, signal 1 is not enough, just like what we discussed in T cell activation. Now there's something called step 3. In step 3, what's going to happen is CD4 ligand interaction happens, and this is a very critical. Okay, CD4 is expressed on the B cell, and CD4 ligand is expressed on the activated CD4 T cell. Okay, so when CD4 binds to CD4 ligand, it triggers class switch recombination. It also causes affinity maturation, germinal center formation, memory B cell development. So what does it do? It causes class switch recombination, affinity maturation, germinal center formation, and memory B cell development. Okay, and this interaction is non-negotiable. Okay, just to recap, what does CD40 ligand enable? Number one, it enables class switching. It causes IgM to go to IgG, IgA, and IgE. Okay, it causes affinity maturation and somatic hypermutation in germinal center, and it also produces high affinity antibody. Okay, so the buzzword is germinal center reaction. Okay, and one another thing that you want to remember is what happens if CD40 ligand is defective. You're going to have something called hyper IgM syndrome, and this is a classic exam immunodeficiency. If CD40 ligand is defective, and that is usually X-linked, B cells cannot class switch, and B cells cannot undergo affinity maturation, and that results in high IgM, low IgG, IgA, and IgE. Okay, so key clinical consequences that happen with this is that there is going to be recurrent pyogenic infections, there's going to be opportunistic infection. That's because macrophage activation via CD40 also fails, and there's no germinal centers in your lymph nodes. Okay, Let's move on. So I would like to close off this lecture by talking about anergy. Okay, so anergy is the state of functional unresponsiveness, and it occurs when lymphocytes encounter antigen without proper co-stimulation, like we saw in our previous slides. This is a major mechanism of peripheral tolerance, and it's also one of your exam's favorite questions. And that concludes our lecture on cellular immunity.